hopefully will support approval in the United States. So these are the 5 COVID-19 vaccines that are either in or recently completed phase 3 efficacy trials. And so these are the 5 vaccines that have the potential of being rolled out in the United States during this year. These are all large phase 3 trials, 30 to 45,000 people. The first 2 we talked about already, RNA vaccines from Pfizer and Moderna. The launch of these trials was in late July, and the efficacy was announced in November, and EUA approved in December. The AstraZeneca vaccine is based on a chimpanzee adenovirus. Earlier studies started in the UK and Brazil, and those were the results that were announced in November. The US-based trial of the AstraZeneca vaccine started in August, and because of some pauses along the way, likely will read out sometime in the spring, maybe March or April. The J&J vaccine is a vaccine that our group has been very involved in, and I'll tell you a lot more about the scientific underpinnings of this vaccine based on a human Ad26 vector. And the launch of this phase 3 trial was in September, and the efficacy results will be announced very soon, essentially this week. This month, in the next, probably in the next 2 weeks. And nobody knows what it's going to show yet. The Novavax vaccine is the first protein-based vaccine candidate to be tested in the US. And this phase 3 study launched just a few weeks ago in December and will probably read out in the springtime. So vaccine development typically takes many years or decades for a new pathogen. So how can we develop a COVID-19 vaccine, or rather multiple COVID-19 vaccines, in approximately 1 year? That is truly unprecedented in the history of science. First, there's a clear global pressing need. And ultimately, it really is the clinical need that has driven so many vaccine developers to move their technologies forward so fast. It's really because of the patients in the end. On a practical level, it's possible to go quickly because the lead vaccine programs use vaccine platforms that have already been tested for other pathogens. In other words, we use adenoviral vectors. We've used them before, so we know a lot about them. Moderna and BioNTech had used RNA vaccines before, for example. Also, development activities can be performed in parallel rather than in series. Rather than the long sequence of steps typically for vaccine development, it's possible to do a lot of things at the same time if you're willing to tolerate programmatic and financial risk. What I mean is, for example, most of the vaccine developers, all the large ones, start to mass produce their vaccines during the context of the phase 3 trials rather than wait until after they're done. Now, that's a big risk because if the trials fail, they'll literally have millions of vials of vaccine that will literally just be thrown in the trash. But it's a calculated risk, and I think you'll probably all agree with me that the companies taking that calculated risk do not pose any additional risk to patient safety. It's just a risk, a programmatic risk that the companies are taking. And I think that's really critical to emphasize that moving vaccine forward quickly and efficiently is actually not being rushed. If anything, there's even more attention to safety than for any other vaccine programs in terms of the size of the studies and the total number of person safety years. But regardless, mass production and global distribution is required because the goal is to achieve herd immunity to end the pandemic. So since January of last year, we've really taken a two-pronged approach. One is to address critical questions of basic research of immunology and correlates that we think are important, not just for our vaccine program, but really for all the vaccine programs. And secondly, was the actual activities to develop our specific vaccine candidate. I'll tell you about both of them today. So 2 questions in the basic science realm for COVID vaccine development in general is first, is there natural protective immunity? Will individuals who recover from COVID-19 be protected against re-exposure? And secondly, is there evidence of vaccine-induced immunity? And if so, what are the immune correlates of protection? We started addressing these questions a year ago, and we thought that it would be beneficial for the field to get rapid answers in addition to awaiting the large-scale clinical trials. So we set up rhesus macaque model of infection, and we addressed these in the non-human primate model. And these were our first two papers that were published back in May. So in the first paper, we developed the rhesus macaque model. We got a vial of virus from the CDC and the repository from the first Washington patient, the index patient in Washington. And we grew up a challenge stock and we inoculated monkeys at 3 different doses, a high dose in group 1, a mid dose in group 2, and a low dose in group 3. 9 out of 9 monkeys got infected. Here are viral loads and bronchoalveolar lavage in the lungs following infection. You can see all 9 animals were infected. Peak virus was around day 2, and then virus was controlled after about 2 weeks. In nasal swabs in the upper respiratory tract, we also got good virus replication that peaked at around day 2 and was controlled by about 3 to 4 weeks. We also looked at pathology in the monkeys. This is 2 and 4 days following challenge, and you can see clear evidence of viral pneumonia. Panels A and B, you can see there's a dense inflammatory infiltrate in the lung parenchyma. In panel C and D, you can see sloughing of epithelial cells and terminal bronchioles. In panel E, you can see a multinucleated giant cell. In panel F, you can see the emergence of hyaline membranes. In panels G and H, you see immunohistochemistry staining for SARS nucleocapsid, showing an infected multinucleated giant cell in G and an infected pneumocyte in H. Panel I is RNA scope analysis, directly demonstrating SARS-CoV-2 RNA in cells in an area of dense inflammation. And J, K, and L are large bronchi showing acute inflammation in J, and RNA scope and immunohistochemistry staining in K and L, demonstrating infection of ciliated bronchial epithelial cells. So the experiment that we did is, this was one of the first demonstrations of natural protective immunity. And what we did is we took the original 9 monkeys that were challenged, and they resolved their virus by day 21. We waited until day 35, and then we re-challenged them. And following re-challenge, we demonstrated there was strong protection. So in this experiment, we were not able to dissect the correlates of the protection, whether it was the antibodies, the T cells, or something else, but we did demonstrate that an initial infection protects very strongly against re-challenge. And this was one of the first demonstrations of natural protective immunity, I believe the first one in primates, and obviously critical for the entire global vaccine effort, because it's much easier to develop vaccines for pathogens for which there is natural protective immunity. In the second paper that we also published in May, we did our first vaccine study using prototype DNA vaccines. Now, these DNA vaccines were not the vaccines that are more potent and are not the vaccines that we took into clinical trials, but we thought that these research tools would give us early insight into protection. And these early vaccines would teach us about immune correlates and what we could learn and what we could look for in some of the later studies that we did. So we looked at DNA vaccines expressing a series of different spike proteins, and we vaccinated animals and challenged them. And these are peak viral loads in the lung and bronchoalveolar lavage. So even though these are early prototype vaccines, the protection was really quite good. And more importantly than seeing protection, we also saw the first evidence of immune correlates, which is shown here, which is that the level of antibodies induced appear to inversely correlate with peak viral loads in BAL or nasal swabs. In other words, the antibodies induced by the vaccine correlated with protection. And we reasoned that if such a correlate can be confirmed in multiple different platforms in monkeys and ultimately is confirmed in humans, then we can have a very powerful tool for future vaccine development efforts. So I'll pivot now to tell you about the actual vaccine candidate that we helped develop and that now is in late phase clinical trials. This is based on a replication incompetent common cold virus, which is adenovirus serotype 26. We initially developed and vectorized Ad26 as part of our HIV research program and our HIV vaccine development program in the early 2000s. And the first paper from our lab in which we vectorized Ad26, essentially made it into a vaccine vector, was a Journal of Virology paper in 2007. At that time, we had no idea how important or useful that would be. So we made Ad26 for our HIV vaccine program. we were working with a small biotech company in Holland at that point in time called Crucell. And, Crucell was purchased by a large pharmaceutical company, Johnson & Johnson, about 8 or 9 years ago. And then at that point in time, around 2010, then our industry partner suddenly went from a small biotech company to a major pharmaceutical company, which obviously brought forward our HIV vaccine program very strongly. And I should say, phase 3 trials with our Ad26 based HIV vaccine are currently in progress as well. I just don't have time to talk about those today. Our first experience using Ad26 for pandemic responsiveness was in 2016, when we developed an Ad26 based vaccine for Zika, which worked beautifully. And I'll show you one piece of data from that study. And then J&J developed Ad26 based vaccines for Ebola and RSV, which provided a lot of conceptual input to the COVID program. Throughout all of these prior programs, Ad26 has been shown to be safe and immunogenic in over 100,000 people to date. And also we have 10 years of experience from the first Ad26 HIV vaccine trials. So this is not a new platform. We have clinical data for over 10 years, at least in some people, and in over 100,000 people, even prior to COVID. The Ad26 Ebola vaccine was actually approved in Europe this past year. So there is regulatory approval for this. And so this is no longer an experimental platform. With Ad26 RSV, there's also pediatric experience in which this vaccine has been dosed down to babies 6 months of age. So if this vaccine for COVID ends up being safe and effective, then age de-escalation into pediatric cohorts should be readily achievable. And obviously that's important for vaccinating the school-age population for all the back-to-school issues that really range from essentially preschools up until high school. College students, of course, would already be in the adult category. Also, for the Ebola program, J&J produced over 2 million doses of this vaccine. And so we know that mass production is possible. And they've developed formulations that don't require freezing because they had to distribute this vaccine to Africa. So we know that mass production is possible and global distribution is feasible. And through a number of these programs, we also showed the potential for a single shot vaccine with no frozen cold chain required, which we think would be useful on a very practical sense for a mass vaccination campaign for COVID-19. So this is one piece of data from our Zika program. This is a phase 1/2 study in which we inoculated 100 people with 2 different doses of the Ad26 Zika vaccine, low dose and high dose, and half the people got a single shot vaccine, the other half the people got a boost at day 57. So the key takeaways from this clinical trial is that virtually everybody who got a single shot vaccine of Ad26 Zika developed neutralizing antibodies. If you got boosted, you had a huge jump in those neutralizing titers, but both single shot vaccine and 2-shot vaccines had neutralizing antibodies that were durable for at least a year. So this showed, at least for Zika, that a single shot vaccine was a possibility to raise neutralizing antibody responses in the majority of people, and those antibodies were durable. And if you wanted a more potent version of the vaccine, then you could always give a boost. So this really formed the conceptual underpinning of the Ad26 COVID vaccine program. So this is what we did. On January 10th of last year, we downloaded the sequence of the virus that was released that day from the Chinese researchers. We started working on the sequence that same night. That was a Friday evening, US time. We started working on it that same day. And over the weekend, we designed synthetic antigens so that on Monday, January 13th, we were in a position to start creating synthetic genes to design our vaccines. By the end of January, we signed a collaboration agreement with our industry partner, J&J, because they, of course, had the technology to manufacture and scale up and deliver. In February, we started studies in preclinical models and mice and rhesus monkeys. And by the end of February, we developed a viral challenge stock. In early March, we started infection studies in small and large animal models to establish models of disease. And we basically settled on the hamster for a small animal model. I don't have time to talk about that today, but I can refer you to some of our publications, as well as large animal models, which we've used primarily the macaques. By the end of May, we published our first 2 papers on natural and vaccine immunity, which I just showed you. And by the end of July, we started our phase 1/2 clinical trials with the Ad26 based COVID vaccine. Also at the end of July, we published Ad26 vaccine protection in the non-human primate model. And 2 months later, in the end of September, we initiated our first phase 3 study, which was a study of the single shot vaccine in 45,000 individuals worldwide. And 2 months later, in November, we started the two-shot version of this study. So we're studying this vaccine both as a single-shot vaccine and a two-shot vaccine. The single-shot vaccine study will read out, or is expected to read out this month in the next 2 weeks, literally. And so we don't know if it's going to work or not. But if this vaccine proves to be safe and effective, then J&J would be in a position to apply for emergency use authorization essentially immediately. And over the course of this year, J&J is committed to producing at least 1 billion doses of vaccine worldwide and distributing it on a nonprofit basis. So we think that the combination of the scale of 1 billion vaccine doses, the potential for a single shot vaccine, and the potential for the lack of a need for sub-zero freezing will be benefits for mass vaccination campaigns, both in the United States as well as throughout the world. So I'll show you a few slides from our paper from July in which we looked at the immunogenicity and protective efficacy of the Ad26-based vaccine in rhesus macaques. We developed Ad26 vectors encoding the SARS-CoV-2 spike protein together with our colleagues at J&J, and we essentially tested not 1, but 7 different versions of this vaccine head-to-head. I'm not going to go through all the different variations, but we basically looked at a wild type and TPA leader sequence, furin cleavage site mutations, the S2 diproline stabilizing mutations, as well as deletions of the cytoplasmic terminus. I'm just going to concentrate on the bottom one that we called S.pp, because this is the vaccine that ultimately went into clinical trials, that has a wild type leader sequence. It's a full-length spike and has a mutation of furin cleavage site knockout and 2 proline stabilizing mutations originally described by Jason McClellan and colleagues to help stabilize the spike protein. We did a study in 52 rhesus macaques, age 10 to 12, mixed gender. At week 0, we did a single shot vaccine with 10 to 11 viral particles of Ad26 vectors, encoding 1 of 7 spike protein variants, 4 to 6 animals per group. So there were a total of 32 vaccinated animals and 20 sham controls. At week 6, we then performed a challenge using our standard model of 10 to the 5 TCID50 of SARS-CoV-2 by the intranasal and intratracheal routes. These are neutralizing antibody titers induced in the animals following single shot vaccination. By week 4, essentially all vaccinated animals showed neutralizing titers. But you can see there were differences, and the highest antibody titers were induced by the SPP vaccine, suggesting that the wild-type leader sequence is better than the TPA leader sequence, the full-length spike is better than a deletion mutant, and the stabilizing mutations were beneficial. This is protection data. This is viral loads in bronchoalveolar lavage post-challenge. The 20 sham control animals all got infected and had high levels of virus, peaking at day 2 and resolving by about day 10. Very similar to what we see in humans. In the SPP vaccinated animals, there was no virus detected at any point in time, resolving by about. If we look at the other 6 vaccines, then these actually also showed relatively good protection with 0 to 2 breakthroughs per group. And the breakthroughs generally were lower in magnitude and shorter in duration than in the sham controls. This is protection in the upper respiratory tract and nasal swabs, which is always a higher bar to achieve than protection in the bronchoalveolar lavage. In nasal swabs, in the sham controls, all animals showed robust virus replication, and peaking on day 2 and being resolved by about day 10 to 14. In the SPP vaccinated animals, 5 out of 6 animals were protected. 1 animal showed a low blip for 2 days. There were 1 to 3 breakthroughs per group in the other vaccine groups, and the breakthroughs generally were a bit higher and longer in duration than what we saw in the bronchoalveolar lavage. So this is a summary of protective efficacy. These are peak viral loads in bronchoalveolar lavage on the left and nasal swabs on the right. The sham controls basically showed about 5.5 logs of virus on average. And there's differing degrees of protection with the different vaccines. But the vaccine that we picked to move forward based on these data is the SPP vaccine shown here that showed complete protection in the lungs and near complete protection in nasal swabs. One aspect of the study we were able to do is we were able to really confirm the immune correlates that we saw in the DNA vaccine paper. So again, here we saw that the neutralizing antibody titers induced by the vaccine inversely correlated with protection, both the antibody titers at week 2 and even more so the antibody titers at week 4. But in addition to the neutralizing titers, we did another analysis together with Galit Alter of the Reagan Institute using her system serology platform. And we were able to look at many different antibody parameters. And we divided animals into 2 groups, the animals that showed complete protection and animals that showed partial protection. And the discriminating factor that divided animals best into these 2 groups was the neutralizing antibody titer. But other antibody functions also were very effective, such as the antibody-dependent NK cell activation shown here and antibody-dependent cellular phagocytosis shown here. But our interpretation is that the neutralizing antibodies are probably the most important ones, but that other antibody functions likely also contribute. We next looked at the anamnestic responses following challenge in these animals. So here we look at the neutralizing antibody titers in the sham control animals on day 14, and they all come up as expected. But in the SPP vaccinated animals, there was really no evidence of increase in antibody titers, and this lack of an anamnestic response is basically a verification of protection. We did see anamnestic responses in the other 6 vaccine groups, at least in many of the animals in the other 6 vaccine groups. So these data suggest that if a vaccine is unable to fully protect, then there will likely be a rapid anamnestic neutralizing antibody response that likely will substantially limit virus replication. We also looked at T-cell responses following challenge, and these are T-cell responses at week 2 following challenge. CD8 responses on the top, CD4 responses on the bottom. In the sham control animals, there's a broad-based T-cell response against spike, nucleocapsid, and NS 6, 7, and 8, as you would expect. But in the SPP vaccinated animals, all we saw were residual responses to spike, and we didn't see any broadening of these responses or any responses to non-vaccine antigens, again, consistent with the idea that these animals really were completely protected. Essentially, no virus replication means no induction of T-cell responses. So this is an orthogonal measure of protection. So from this study, we demonstrated that a single shot of Ad26 SPP vaccine resulted in complete protection in the lung and near complete protection in the upper respiratory tract. The lack of anamnestic responses suggested minimal to no virus replication in these animals. As we've talked about, we think that a single shot vaccine would have major advantages over a 2-shot vaccine for mass vaccination and pandemic control. But we also know a second shot of the vaccine likely will increase neutralizing titers substantially. So clinically, we think it's important for both a single shot vaccine and a 2-shot version of the vaccine. We also showed that the neutralizing antibody titers were the strongest correlate of protection, consistent with our prior DNA vaccine study. And if you take this study with the previous study together, it's 87 primates, which is starting to get to non-trivial numbers. So the development plans for the Ad26 SPP vaccine. First, it was renamed Ad26 CoV2S, which is the name you might see moving forward. Phase 1/2 studies were started with J&J at the end of July, and the first phase 3 efficacy study was started on September 21st. So I'll talk about one more basic research study and then tell you more about the clinical vaccine program. So immune correlates of protection we think are important to define because immune correlates will contribute to our understanding of viral pathogenesis and immunity. But also defining immune correlates might have some practical value because it's likely that future approvals of vaccines are going to need to be approved based on immune correlates, particularly because it is very likely that placebo-controlled efficacy trials will soon become impossible to do. Essentially, as soon as vaccine availability becomes widespread, then it's unethical and impractical to do a placebo-controlled efficacy trial, which means that vaccine research will stop and that we will not be able to test potentially next generation or improved vaccines moving forward, which of course would not be good. So future approvals of vaccines will likely need to be done based on immune correlates if a convincing correlate can indeed be defined. Also, immune correlates could be useful for understanding the waning of protective immunity following vaccination or infection. So we published this paper about a month ago in which we tried to define immune correlates of protection against SARS-CoV-2 in rhesus macaques. So what we did is we went with our best hunch, which was from the vaccine studies, that antibodies correlated with protection. Also, of course, work from many, many other groups that would lead to similar suggestions. But no one actually has proven that antibody responses protect. To do so, you would have to do an adoptive transfer study, which is what we did for this paper. So in this paper, we aim to determine if purified IgG from convalescent macaques could protect naive animals from infection, and if so, could we define neutralizing antibody thresholds for protection? So we purified IgG from 12 rhesus macaques, from a cohort of convalescent re-challenged macaques that had high titers to start with, and then we adoptively transferred that into 12 naive rhesus macaques. 3 got a high dose of 250 mg per kg, 3 got 25 mg per kg, 3 got 2.5 mg per kg, and 3 got sham controls. And then 3 days later, on day 0, we did our standard 10 to the 5 TCID50 SARS-CoV-2 challenge by the intranasal and intratracheal routes. So these are neutralizing titers following adoptive transfer. At day -3, these are all baseline. So on day 0 is a day of challenge. This represents 3 days following infusion. In the 250 mg per kg group, these animals had neutralizing titers of about 500, and then these declined over time over the 2 weeks following challenge. For the animals that received 25 mg per kg, the titers were almost 10-fold lower, almost exactly mathematically 10-fold lower, about 50. And these titers also declined over time, although one of these animals appears to tick up on day 14. In the animals that received 2.5 mg per kg, these titers were undetectable on day 0, and all of these come up very strongly by day 14, very similar to sham controls. So here's the protection data in bronchoalveolar lavage. The high dose, completely protected. The middle dose, there was 1 breakthrough. The low dose, all 3 animals were infected, but generally showed a shorter duration of virus replication compared to the sham controls. In the nasal swabs, we saw a very similar picture. The top dose protected all 3 animals. The middle dose, there was 1 breakthrough. The lower dose, all animals were infected, but generally had lower peak of virus and shorter duration of virus replication compared with the sham controls. So with Galit Alter and Doug Laufenberger, we also did a logistic regression analysis to define the neutralizing antibody threshold for protection. And in this model, the threshold for protection was about 50, which was relatively low. So a relatively low level of neutralizing antibodies appear to protect. And we said in our paper that these titers should be readily achievable by vaccination. And I should mention that this paper was submitted prior to the announcement of the Pfizer results. We did one more study in this paper, which was a CD8 depletion study. We wanted to ask whether T cells were important for protection. Now, I just showed you that antibodies alone can protect. So what do T cells have to do with it then? So we used a different model to ask the question of T cells. Rather than an adoptive transfer into naive animals, we used convalescent macaques. We waited for a while to watch their antibody titers wane. And in the setting of borderline waning antibody titers, we then did a CD8 depletion study to determine the role of CD8 responses. So what we did is we had 10 convalescent animals. We depleted 5 of them with a CD8 monoclonal antibody against CD8 alpha. We did similar studies with CD8 beta antibody, and 5 animals were not depleted. And then 3 days later, we did a re-challenge with 10 to the 5 TCID50 of SARS-CoV-2. So this is the relevance of CD8 responses in protection against re-challenge. So this just shows the waning neutralizing titers. In the re-challenge animals, that was the source of the adoptive transfer study shown here at very high levels. After a first challenge at week 4, then here are the levels of just over 100. And 3 weeks later, then many of these had declined to less than 100. So these are the animals we used. These 10 animals are the ones we used in the CD8 depletion study. So we depleted CD8 cells and then we re-challenged, and here's the protection data. In bronchoalveolar lavage, there was pretty good protection in both groups, although there was 1 blip in the CD8 depleted antibodies, CD8 depleted animals. In nasal swabs, we saw more of a profound effect. There was 1 blip in the sham controls, but all 5 out of 5 animals showed viral breakthrough following CD8 depletion. And this just shows this just shows that this increase in peak viral loads following re-challenge is statistically significant with CD8 depletion. Now, it's not as high as in naive animals, so it's clear that other factors also help control virus in these, but it was also clear that the CD8 depletion had an impact. So in this study, we basically showed that purified IgG can protect macaques against SARS-CoV-2 challenge in a dose-dependent fashion. We also showed that the threshold neutralizing antibody titer for protection in this model is low at around 50, and these titers should be readily achievable by vaccination. We also showed that CD8 depletion reduced protection against re-challenge in convalescent macaques with waning antibody titers. And these data suggest that antibodies alone can protect, but that cellular responses likely contribute when antibody titers are borderline or subprotective. So I'll turn now to the clinical program, and the phase 1/2 study was called COVID-19, which is a randomized double-blind placebo-controlled trial of the Ad26-CoV-2S vaccine. These interim results were published at the end of September in medRxiv, and there's actually an update of this data that's going to be published in the New England Journal of Medicine coming out either today or tomorrow, but basically shows the same findings as was reported previously. And here we show that if you just look at cohort 1A, this is designed just like the Zika trial I showed you earlier. There's 2 doses of the vaccine, and half get a single shot vaccine and half get boosted in a placebo group. Don't worry about the rest of it. Just remember that cohort 3 is similar to cohort 1, except cohort 3 is elderly adults over 65 with no upper age limit. So these are binding antibodies in humans following vaccination. This is a placebo-controlled trial. So group unblinded, the placebo recipients had negligible responses. And the vast majority of individuals developed binding antibody titers within 4 weeks of vaccination. This is true in the younger adults and in this pilot sample in older adults. And with a full cohort of older adults, it looks very similar. So for neutralizing antibodies, we saw a very similar phenomenon that there were neutralizing antibodies induced by both dose groups in both cohort 1 as well as in cohort 3. So this vaccine, after a single shot, raised binding and neutralizing antibodies in the vast majority of individuals. I think there was only 1 that wasn't. And interestingly, when you take the data beyond this, then these responses actually continue to go up between week 4 and week 10 or 12. These are CD4 T cell responses. So the vaccine also induced cellular responses in nearly all individuals. These are CD4 T cell responses that are exclusively of the Th1 flavor rather than the Th2 flavor, as well as CD8 T cell responses shown here. So in terms of the phase 3 program, we started this phase 3 study as quickly as we could after the phase 1/2 study. It was exactly 2 months because we had to wait for enrollment of the phase 1/2 study, and then we had to get the week 4 data in. But we were able to start the first phase 3 study at the end of September. This is COVID-3001, a phase 3 randomized double-blind placebo-controlled trial of the efficacy and safety of Ad26-CoV-2S. As of December 17th, this study has been fully enrolled of 45,000 individuals, and we do expect results this month. I personally have no clue whether this vaccine is going to work or not at this point in time. It's a 1-to-1 vaccine placebo randomization. This is testing a single shot vaccine at 5 times 10 to the 10th viral particles and no requirement for a frozen cold chain. This vaccine is being studied in the US, South Africa, Brazil, and multiple countries in Latin America. The second phase 3 study that's underway is called COVID-3009. This study started in November, so this study is still enrolling. Target enrollment is 30,000, 1-to-1 placebo vaccine randomization, and it's basically the same vaccine, but people get a boost after 2 months. And this study is being conducted in the US, in the UK, and many countries in Europe, as well as several others. So I'll close with this slide about my own perspectives of COVID-19 vaccines. Vaccine development for COVID-19 is proceeding faster than for any pathogen in history. Safety and public trust are critical. because a vaccine that's safe and effective but is not used will fail in its mission to stop the pandemic. I believe that multiple vaccines need to be implemented in parallel, since it's not clear at this point which vaccines will be the most durable and also the most deployable. And regardless, we just simply need more vaccines. We need to accelerate the vaccine pace in this country and the world. We believe that preclinical challenge models may provide useful insights into vaccine efficacy, correlates, and pathogenesis. And finally, we have to realize that there are over 7 billion people in the world. So we need not one or two, but we actually need multiple vaccines to be successful and simultaneously deployed at scale to end the pandemic. No one company, no one vaccine will be able to achieve this. I'll stop there, and here are the acknowledgements for the Ad26 vaccine study in non-human primates and the adoptive transfer studies, and I apologize I don't have a slide for the clinical team. But this is our lab. This is an old picture. It was taken over a year ago, and so this is somewhat out of date, but we hope that we will be able to get back to a state where we can take pictures like this, hopefully sometime before the end of this year. So I'll thank you for your attention. Happy to take any questions.
Yes, I sort of have two questions. One is, although the second is a bit more specific to like proteins and stuff. Okay, so the first question, there was a slide that was titled SARS-CoV-2 infection protects against rechallenge in rhesus monkeys. So is that generally applicable to humans as well? The general trend that the second infection is not as virulent as the first time around?
You know, I mean, there's too little data in humans. They're case reports.
Okay.
And the general perception is probably if you did get re-exposed,